third-party content has been copied and communicated pursuant to Part 5A or 5B of the Copyright Act, unless indicated otherwise.
Let's begin by looking at a diagram of what happens at a chemical synapse. Now, there are several basic steps involved in every chemical synapse, and understanding the chemical events at a synapse is fundamental to understanding the functioning of the nervous system. Each of these steps is a biochemical event that can potentially be influenced in some way, thus altering function. And most of the effects of drug action, which we'll look at later, occurs at one of these steps. The first step is the synthesis step. The neuron makes the chemicals that serve as neurotransmitters. It synthesizes the smaller neurotransmitters in the axon terminals and the larger peptide molecules in the cell body. The neuron synthesizes its neurotransmitters from precursor molecules derived from foods. Acetylcholine, which we'll look at later, for example, is synthesized from choline, which we find in cauliflower and milk. The body can also make choline from lecithin, a component of egg yolks, butter and peanuts. Serotonin, another major neurotransmitter, for example, is synthesized from tryptophan, which is found to be high in soy, for example. The second step is the transportation and release. So certain neurotransmitters, such as acetylcholine, are synthesized in the presynaptic terminal, close to where they are released. However, the larger neurotransmitters, including the peptides, are synthesized in the cell body and transported from there down the axon to the terminal. Action potentials also travel down the axon. The speed of transportation varies from only 1 millimeter per day in thin axons to more than 100 millimeters per day in thicker ones. Even at the highest speeds, though, transport from cell body to terminal may take hours or days in the longest axons. As for the actual release element, at the presynaptic terminal, an action potential causes calcium to enter the cell, thereby evoking the release of the neurotransmitter from the terminals and into the synaptic cleft, that space between the presynaptic and the postsynaptic neurons. When an action potential reaches the end of the axon, the depolarization changes the voltage across the membrane and opens voltage-dependent calcium gates in the presynaptic terminal. As calcium flows through specialized channels into the presynaptic terminal, it causes the neuron to excrete neurotransmitter through its membrane and into the synaptic cleft. Now, this process of excretion, where the neurotransmitter is released, is quick, lasting 1 or 2 milliseconds. The third stage is what we call receptor interaction. The released transmitter crosses the synaptic cleft and binds weakly to specialized receptors on the postsynaptic membrane, where it initiates an action potential, which is a brief, violent electrical pulse. If the neurotransmitter is released at an excitatory chemical synapse, it induces an action potential in the postsynaptic element. If the neurotransmitter is released at an inhibitory chemical synapse, it stops the neuron from firing in the postsynaptic element. At an excitatory chemical synapse, a single synapse cannot force the postsynaptic cell to produce an action potential. Rather, to trigger a nerve impulse, many synaptic influences are summed. Let me just change figures to show you what happens in terms of summation. Now, there's two kinds of summation, what we call temporal summation and spatial summation. Temporal summation occurs when the postsynaptic membrane receives several impulses from one presynaptic neuron over a period of time, hence temporal, as in time. Spatial summation occurs when the postsynaptic membrane receives impulses from several presynaptic neurons at the same time. Now, temporal summation and spatial summation pretty much occur together. So that is that a neuron might receive input from several axons at approximately, but not exactly, the same time. Now let's return to our chemical synapse figure and back to stage 3, where the chemical's been released, or the neurotransmitter's been released, and it binds to the receptors. The released neurotransmitter crosses the synaptic cleft and binds to specialized receptors on the postsynaptic membrane. The receptors are proteins embedded in the postsynaptic membrane. When the neurotransmitter attaches to the active site of the receptor can directly open a channel, what's known as an ionotropic effect, or it can exert a slower but longer lasting effect known as a metabotropic effect. Ionotropic effects are almost immediate. They emerge about 10 milliseconds after the release of the neurotransmitter and last about 20 milliseconds. Ionotropic effects are rapid and short-lived. Ionotropic synapses are useful for conveying information about visual and auditory stimulation, muscle movements, and other rapidly changing events. For example, acetylcholine exerts ionotropic effects at some of its synapses. Now, metabotropic effects are slower and longer lasting in comparison than ionotropic effects, and metabotropic effects emerge 30 milliseconds or more after the release of the neurotransmitter and last seconds or minutes or even hours. The fourth stage is the separation from the receptors. A neurotransmitter does not normally linger at the postsynaptic membrane. If it did, it might excite or inhibit the postsynaptic neuron indefinitely. So various neurotransmitters are inactivated. That is, the neurotransmitter is converted into an inactive chemical in the area of the synaptic cleft. Acetylcholine, for example, after it activates a receptor, is broken down into two inactive fragments, acetate and choline. The choline diffuses back to the presynaptic membrane, which takes it up and reconnects it with acetate already in the cell to form acetylcholine again. The next stage, or the fifth stage, is known as reuptake. In other synapses, the neurotransmitter is taken back up into the presynaptic neuron for recycling. Serotonin and dopamine, for example, are not broken down into inactive fragments at the postsynaptic membrane, but simply detached from the receptor. The presynaptic neuron takes up most of these neurotransmitter molecules intact and reuses them. This process is called reuptake, and you'll find that in some synapses both inactivation and reuptake may occur to remove the transmitter from the synapse.